Okay, welcome back to Biology in Focus. This is Chapter 36, Reproduction and Development. My name is Mr. Sparks, and I will be guiding you through this lecture for John Tyler Community College. Okay, so today we're going to talk about sexual reproduction and development of the embryo. The overview has to do with the sea slug. The sea slug produces sperm and eggs and acts as a mother and a father to the next generation. They're known as hermaphrodites. Animal reproduction takes many forms. We're going to discuss some of those forms throughout this chapter. And a population outlives its members only by reproduction. So the generation of new individuals from existing ones. Here is a picture of two nudibranchs mating. The name nudibranch means naked lung, and you can see the gills on their lower back right here. That's the structure that gives them their names. Here is their mating apparatus, and each nudibranch is both male and female. They are hermaphrodites, and this species will the sperm will fertilize the eggs in this one, and this one sperm will fertilize the eggs in this one, and then they'll bear offspring in a few days, and in a few weeks those eggs will hatch. Both asexual and sexual reproduction occur in the animal kingdom. Sexual reproduction is the creation of an offspring by the fusion of the male gamete, the sperm, and the female gamete, the egg, to form a zygote. Asexual reproduction is the creation of offspring without the fusion of egg and sperm. So now we're going to talk about some different forms of asexual reproduction. Many invertebrates reproduce asexually by budding, in which new individuals arise from outgrowths of existing ones. These are like hydras or certain other cnidarians, jellyfish. They can have a growth on the outside of their. They have a little growth that arises on the outside of their bodies, and those little growths can bud off and become separate individuals. Also common among invertebrates is fission, the separation of a parent organism into two equally sized individuals. Here is the fission of a sea anemone. Here it is. It's fissioning into two separate organisms. Fragmentation is the breaking off of body into pieces, some of which may develop into adults. Starfishes are notorious for this type of reproduction. In fact, the crown of thorn starfish was destroying the coral reef in Australia, and when fishermen would catch the crown of thorn starfish in their nets, they'd cut them up, and they were trying to destroy them, but actually what they were doing is causing them to reproduce. So every leg of the starfish if the starfish has 5 legs and you cut it into 5 pieces, each leg would turn into a new starfish. Fragmentation may be accompanied by regeneration, the regrowth of lost body parts. Parthenogenesis is the development of an individual from an unfertilized egg. And interestingly enough, this parthenogenesis does occur in some higher vertebrates. Sexual females have half as many daughters as asexual females. This is the twofold cost of sexual reproduction. Despite this, almost all eukaryotic species reproduce sexually. So even though asexual reproduction produces more offspring, sexual reproduction is the preferred mode of reproduction in most eukaryotic species, because it provides for genetic diversity in the offspring, and with that genetic diversity, you can through evolutionary pathways, you can experience and deal with different environmental stresses. Okay, here's asexual reproduction. Each female produces offspring. You get 4 times as many in sexual reproduction. In this schematic, the one female has 2 offspring. One is a male. That male does not reproduce. Only the female reproduces. Okay, so you get the idea. So you get greater numbers through asexual reproduction, but you get greater genetic diversity through sexual reproduction. Sexual reproduction results in genetic recombination. The resulting increased variation among offspring may enhance the reproductive success of parents in changing environments. Most animals exhibit reproductive cycles related to changing seasons. Reproductive cycles are controlled by hormones and environmental clues. Because seasonal temperature is often an important cue in reproduction, climate change can decrease reproductive success. Okay, for example, here is as a result of global warming and climate change, the number of caribou offspring in West Greenland study site has fallen 4-fold. The reason for this is that the arrival of caribou at the calving grounds coincided with a brief period during which the plants were nutritious and digestible. From 1993 to 2006, during the period of this study, however, average spring temperatures in the calving grounds increased by 4 degrees Celsius, and the plants now sprouted 2 weeks earlier. Because the caribou migration is triggered by day length and not temperature, there is a mismatch between the timing of the new plant growth and the caribou birthing. Without adequate nutrition from the nursing females, production of caribou offspring has declined by 75% since 1993. In some species of whiptail lizards, Aspidocellus, the members of the breeding pairs alternate roles. Reproduction is exclusively asexual, and there are no males. However, the courtship and mating behaviors still play an important role, with one female of each pair mimicking the male mating behavior. The two alternate roles 2 or 3 times during a breeding season. So here the Aspidocellus, there are only females in this population, and here is one female that's has the behavior of the male during a certain hormonal cycle, and the other female has the behavior of the female, and she is carrying the egg, and the egg is parthenogen. It's not fertilized. It's like the process is called parthenogenesis, and it becomes viable, and then she lays the egg, and then that's how they perpetuate the species. Okay, so here's the close-up picture, and here's the diagram of here is one lizard. The red lizard has got estradiol released into her system. The ovary size is increasing. Ovulation peaks at a given point. During that given point, the lizard that plays the male role has a spike in progesterone levels, and then those progesterone levels fall, and then the estradiol levels peak in the other one again. And okay, so these two alternate roles. Okay, one is the sexual behavior is in correlation with the cycle of ovulation. So a different reproductive cycle occurs in some species of whiptail lizards, the genus Aspidocellus, in which members of the breeding pair alternate roles. In these species, reproduction is exclusively asexual. There are no males. Nevertheless, these lizards have courtship and mating behaviors very similar to the sexual species of Aspergillus genera. During the breeding season, one female of each mating pair mimics the male. Each member of the pair alternates roles 2 or 3 times during the season. An individual adopts a female behavior prior to ovulation when the level of the female sex hormone estradiol is high, and then switches to the male-like behavior after ovulation when the level of progesterone is high. Variation of patterns in sexual reproduction. For many animals, finding a partner for sexual reproduction may be challenging. One solution is hermaphrodism, like the sea slugs, the nudibranchs, in which each individual has a male and female reproductive system. Any 2 hermaphrodites can mate, and some hermaphrodites can self-fertilize. These occur mostly in the invertebrate fauna. Individuals of some species undergo sex reversals. For example, in coral reef fish, the blue-headed wrasse is a lone male defines a harem of females. If that male dies, then the largest female in the group transforms into a male. This is an actual phenomenon that's been observed many times. Within a few weeks, this individual can begin to produce sperm instead of eggs. So they actually change gender in the mid-course of their lives. Sexual reproduction requires fertilization, the union of sperm and eggs. In external fertilization, the eggs shed by the female are fertilized by the sperm in the external environment. Usually this occurs with amphibians and species that live closely associated with water, like fish and amphibians. In internal fertilization, sperm are deposited near the female reproductive tract, and fertilization occurs within that tract. So this is like all birds and reptiles and mammals. A moist habitat is almost always required for external fertilization to prevent the gametes from drying out and to allow the sperm to swim to eggs. Many aquatic invertebrates simply shed gametes into their surrounding water, like coral and certain polychaete worms. There will be certain periods of time based on lunar cycle and tidal cycles. These invertebrates will just release sperm and egg into the environment, and then fertilization will occur at random. In this case, the timing and release of the gametes is crucial to ensure that sperm and egg encounter one another. If external fertilization is not synchronous across the population, courtship behaviors may lead to fertilization. Okay, here are these. Looks like, I don't think it's a bullfrog, but it's closely related species. Many species of amphibians reproduce by external fertilization. In most of these species, behavioral adaptations ensure that a male is present when the female releases her eggs. Here, a female frog on the bottom has released a mass of eggs in response to being clasped by a female. The male released sperm, not visible here, at the same time, and the external fertilization has already occurred in the water. So the male frog is just right on top of the female, and he's trying to get his sperm to fertilize those eggs to increase his reproductive fitness. Okay, now all of these eggs here are from this one frog, and they just become a big gelatinous fertilized mass. You probably have seen masses of salamander or frog eggs in ponds as you explore the woods. And okay, so what happens is that these species, they don't have a lot of parental care, so they produce a lot of offspring. So in the lack of parental care, you need to have a lot of offspring to increase the chances of those individuals surviving. Because these are just, like, they're great food. They're highly nutritious. Other animals will find these food, like fish or whatever, and they'll gobble them up. And when the little tadpoles hatch out, those tadpoles are also highly vulnerable. However, fertilization occurs when mating animals may use pheromones, chemicals released by the one organism that can influence the physiology and behavior of another individual of the same species, ensuring the survival of offspring. Internal fertilization is typically associated with production of fewer gametes, but the survival of a higher fraction of zygotes. Internal fertilization is often associated with mechanisms to provide production of embryos and parental care of young. The embryos of some terrestrial animals develop in eggs with calcium and protein containing shells and several internal membranes, like particularly reptiles and birds. And some reptiles will take care of their young fairly consistently, like crocodiles, for example. But turtles will just lay a nest and they'll depart, and then those turtle eggs will hatch and they'll be on their own. Birds, of course, are well known for taking care of their offspring. Bird eggs hatch in a nest and they're taken care of in the nest. The parents bring them food until they fledge and become their own bird. Some other animals retain the embryo, which develops inside the female. In many animals, okay, so now we're talking about mammals and primarily. In many animals, parental care helps ensure survival of offspring. Okay, here's a water bug or water boatman. It's a little insect that lives in freshwater streams and ponds, and it actually carries its eggs on its back. Compared with many other insects, the giant water bugs of the genera Abetus and Belostoma produce relatively few offspring, but they have much greater parental protection. Following internal fertilization, the female glues her fertilized eggs onto the back of the male, shown here. The male carries them for days, frequently fanning water over them to keep the eggs moist and free of parasites. Reproductive organs produce and transport gametes. Sexual reproduction in animals relies on a set of cells that are precursors for eggs and sperms. Variation in reproductive systems. Many, but not all, of animals have gonads. These are organs that produce gametes. Most simple systems do not have gonads, but gametes form in undifferentiated tissues. They're like we're talking about invertebrates there. So when we're talking about most vertebrates have gonads. More elaborate systems include sets of accessory tubes and glands that carry, nourish, and protect the gametes, and sometimes developing embryos. Most insects have separate sexes with complex reproductive systems. In many insects, the female has a spermatotheca in which sperm is stored during copulation. So many insects are able to have sexual relations, and then they store that sperm in a special container or a special region called the spermatotheca, and there it's saved until the insect is properly receptive, and then it's the female can then use the sperm at her according to her needs. A cloaca is a common opening between external environment and the digestive and excretory and reproductive systems. This is basically all fish, all amphibians, all reptiles, and all birds have the cloaca. It's the common opening to the external environment. It's where they defecate, and they also receive, the female receives the sperm from the male. Okay, all that happens in the cloaca. It's a single opening. The cloaca is common in non-mammalian vertebrates. Mammals that have a separate opening to the mammals, however, usually have a separate opening to the digestive tract. The male's external reproductive organs are the scrotum and the penis. The internal organs are gonads that produce sperm and reproductive hormones, accessory glands that secrete products essential to sperm movement, and the ducts that carry sperm and glandular secretions. Okay, here we have a diagram of the male reproductive anatomy. Here are the testes. This is where the sperm is produced inside what's called the seminiferous tubules. The seminiferous tubules produce the sperm, and then the sperm are transported to the epididymis. Inside the epididymis, there's about 6 meters of fine tubules in here. It takes about It takes about 3 weeks for the sperm to travel through here. But by the time they end here, they are completely motile. They are fully formed, and they travel into the vas deferens. From the vas deferens, they travel around, circulate over the bladder, and in past the seminal vesicule. For each testes has a separate system like this. So there are 2 seminal vesicles. The seminal vesicles produce about 60% of the volume of semen. The fluid from the seminal vesicles is thick, yellowish, and alkaline. It contains mucus, the sugar fructose, which provides most of the sperm's energy, a coagulating enzyme, ascorbic acid, and local regulators called prostaglandins. Okay, now traveling down the vas deferens from here, we have we move into the ejaculatory duct through the prostate gland. The prostate gland secretes its products into the urethra through small ducts. This fluid is thin and milky. It contains anticoagulant enzymes and citrate, a sperm nutrient. The gland undergoes benign, non-cancerous enlargement in more than half of all men over age 40, and in almost all men over 70. In addition, prostate cancer which most afflicts men 65 and older, is one of the most common human glands. Okay, another important gland is the bulbourethral gland. The bulbourethral glands are a pair of small glands along the urethra below the prostate. Before ejaculation, they secrete clear mucus that neutralizes any acidic urine remaining in the urethra. The bulbourethral fluid then carries some of the sperm released before ejaculation, which is one reason for the high failure rate from the withdrawal method for birth control. That's known as coitus interruptus. So always there could be a little bit of active live spermatozoa traveling here. Okay, so the penis has like 3 spongy tissues in it, known as erectile tissue. They become filled with blood, and the penis becomes erect during sexual arousal. And this allows the penis to enter into the vagina, and then you all know what happens then. Okay, I'm not going to go over this too much. I think you guys you're in college, you're probably familiar with most of this. We'll go over some of the details in the lecture here shortly, but this is just a diagram for you to be familiar with. We want to have one for the female reproductive system we're going to go over in shortly. The male gonads or testes produce sperm in highly coiled tubes called seminiferous tubules. The production of normal sperm cannot occur at the body temperatures of most mammals. The scrotum is a fold of the body wall of a sac of skin, and it maintains the temperature at 2 degrees Celsius below the core temperature of the body. So on hot days, the scrotum relaxes and on cool days the scrotum contracts. And all of this is to modify the temperature of the testes so that they have optimal temperature for the production of sperm. From the seminiferous tubules of the testes, the sperm passes into a coiled duct of the epididymis. During ejaculation, sperm are propelled through the muscular vas deferens and the ejaculatory duct, and then exit the penis through the urethra. Semen is composed of sperm plus secretions from 3 sets of accessory glands. The 2 seminal vesicles contribute about 60% of the total volume of semen. The prostate gland secretes its products directly into the urethra through several small ducts. Bulbourethral glands secrete a clear mucus before ejaculation that neutralizes acidic urine remaining in the urethra. The human penis is composed of 3 cylinders of spongy erectile tissue. During sexual arousal, erectile tissue fills with blood from the arteries, causing an erection. The head of the penis or glans has a thinner skin covering the shaft. The prepuce or foreskin is a fold of skin covering the glans. It is removed when the male is circumcised. Human female reproductive anatomy. The female external reproduction structure includes a clitoris and 2 sets of labia. The internal organs are a pair of gonads and a system of ducts and chambers that carries the gametes and house the embryo and the fetus. Okay, so in the female reproductive system, we have the vaginal opening where the penis enters, and then ejaculation occurs and travels into the uterus, and fertilization of the egg usually happens in the fallopian tube, or what they call here the oviduct. Eggs are released from the ovary. They're kind of, eggs are like, they're like in interstitial fluid for a moment in time, and then they travel into the oviduct, and then they travel down here. They might get fertilized about here. Infrequently, an egg can get stuck in here and cause what's called an ectopic pregnancy. This is when the fetus develops in the fallopian tube, but normally the fertilization occurs here, and then the egg will travel down into the uterus, where the uterine lining is generally thickened on a monthly basis. And if the egg is fertilized and the conditions are proper in the uterus, the egg will implant into the wall of the uterus and become a zygote, and then a blastula, and then an embryo. Okay, so here's the cervix. It's the opening of the uterus, and these are the major parts of the female anatomy. Okay, the labia majora, labia minora, and all these are the body, the glands, and the prepuce are all parts of the clitoris. Okay. The female gonads are a pair of ovaries which lie in the abdominal cavity. Each ovary contains many follicles which consist of partially developed eggs called an oocyte, which are surrounded by support cells. Upon ovulation, a mature egg is released. It travels from the ovary to the uterus via the oviduct. Cilia in the oviduct convey the egg to the uterus, also called the womb. The uterus lining, the endometrium, has many blood vessels. The neck of the uterus is the cervix, which opens into the vagina. The vagina is a muscular but elastic chamber that is the repository for sperm during copulation, and serves as the birth canal. The vagina opens to the outside of the vulva, which consists of the labia majora, the labia minora, the hymen, and the clitoris. the mammary glands are not part of the reproductive system, but since they are an important part of reproduction, we're going to include them in parts of this lecture. Within the glands, there are small sacs of epithelial tissue that secrete milk. Gametogenesis is the production of gametes. There is a close relationship between the structure and function of the gonads. Okay, here's the overview. Here are the testes. Here inside the testes are the seminiferous tubules. They are producing the sperm. The sperm there's spermatogenesis includes diploid mitosis until it reaches the meiosis phase, and then those meiotic individuals then become the sperm. So out of every primordial sperm germ cell, there are ultimately 4 sperms. So males make a lot of gametes constantly. That's part of the male strategy. Okay, inside the lumen, the interior area of the seminiferous tubule, this is where the sperm are enclosed. And here's a schematic of the sperm. It's got the acrosome, the nucleus, the mitochondria. Okay, we're going to talk about this. There's a graphic in greater detail after this slide. Okay, so here is the testes, and inside the testes there are seminiferous tubules that are wrapped up really tightly. These seminiferous tubules dump down into this duct which travels to the epididymis. The epididymis has 6 meters worth of tubules, and that this is where it takes 3 weeks for the sperm to move from the beginning of the epididymis to the end of the epididymis and into the vas deferens here. Okay, so this is about the spermogenesis. Okay, so take a closer look inside here of the seminiferous tubule, and we have the spermatogonia. The male is producing these all the time. It produce the primary spermatocyte, the secondary spermatocyte. This is where meiosis begins to occur. Okay, spermatids in 2 different stages. These are spermatids that are not flagellated and they're not mature yet. And then here are the mature sperm cells. And these sperm cells then are taken up by the epididymis, where they finish their maturation, and at the end of their maturation they travel into the vas deferens. Okay, here's the schematic of the production of the sperm cells. There's a spermatogonial stem cell, spermatogonium derived from this, and then primary spermatocytes. So this divides into 2, so there's 2 primary spermatocytes, and then there's meiosis, and each one of these primary spermatocytes becomes 4 sperms. Okay, here's the schematic of the individual sperm. There is the flagella, and this numerous mitochondria are inside that help to run the flagella to propel it towards the eggs. And here's the acrosome. The acrosome is a body of enzymes that helps to penetrate into the membrane of the egg, where the egg will then become fertilized by the nucleus. Okay, so there are just hundreds of thousands of these little guys running around. Okay, in females, females basically have, it's interesting because at birth the ovaries contain about 1 to 2 million primary oocytes, of which about 500 fully mature between puberty and menopause. To the best of our common knowledge, women are born with all the primary oocytes that they will ever have. Oogenesis begins in female embryo with the production of oogonia from the primordial germ cells. The oogonia divide the oogonia divide by mitosis to form cells that begin meiosis, but stop at process prophase 1 before birth. These developmentally arrested cells, called primary oocytes, each reside within the small follicle, a cavity lined with protective cells. Beginning at puberty, follicle-stimulating hormone periodically stimulates a group of follicles to resume growth and development. Cells of the follicle produce primary female sex hormones, estradiol, a type of estrogen. Typically, only one follicle fully matures each month, with its primary oocyte completing meiosis 1. The second meiotic division begins but stops at metaphase. Thus arrested in meiosis 2, the secondary oocyte is released at ovulation, when the follicle breaks open. Only if a sperm penetrates the oocyte does meiosis 2 resume. In other animals, the sperm may enter the oocyte at the same stage, earlier or later. Each of the two meiotic divisions involves unequal cytokinesis, with the smaller cells becoming polar bodies that eventually degenerate. The first polar body may or may not divide again. Thus, the functional product of a complete oogenesis is a single mature egg already containing a sperm head. Fertilization is defined directly as the fusion of the haploid nuclei of the sperm and the secondary oocyte, although we often use it loosely to mean the entry of the sperm of the head into the egg. The ruptured follicle left behind after ovulation develops into a mass called the corpus luteum. The corpus luteum is also known as the yellow body. It secretes additional estradiol as well as progesterone, a hormone that helps maintain the uterine lining during pregnancy. If the egg is not fertilized, the corpus luteum degenerates and follicle matures during the next cycle. At birth, the ovaries together contain 1 to 2 million primary oocytes, of which about 500 fully mature between puberty and menopause. But the best of our knowledge, women are born with all the primary oocytes they will ever have. It's worth noting, okay, so basically that's the summary of oogenesis. Okay, every female is born with the total number of primary oocytes that she will ever have. Of those, about a quarter will, or about 500 will fully mature. Those that do fully mature, they become these secondary oocytes that are they're basically brought up from storage banks, if you will, brought up from storage banks to the surface of the ovary, and where they mature in the follicle, and then they are released. The egg is released from the follicle, and then it's taken up by the fallopian tubes. Looks kind of like an upside down palm tree. We saw the fallopian tubes, in one of the previous slides, and then that ruptured follicle sort of contracts and becomes the corpus luteum, where it governs the production of certain hormones that are necessary to maintain the uterine lining during pregnancy. Okay, so here is the ovary, and there's the little oocyte, the primary oocyte, growing follicle, the mature follicle. There's the egg. The egg ruptures out of the follicle, and then it goes into the uterus, into the fallopian tube, and then ultimately makes its way towards the uterus. If the egg is not fertilized, then it's evacuated by the body along with the lining of the uterus, and that is the blood flow of menstruation. Spermatogenesis is the development of sperm, is continuous and prolific. Millions of sperm are produced per day. Each sperm takes about 7 weeks to develop. Oogenesis is the development of a mature egg, is a prolonged process. Immature eggs form in the female embryo but do not complete their development until years or decades later. Spermatogenesis differs from oogenesis in 3 ways. All 4 products of meiosis develop into sperm, where only one of the 4 becomes an egg. Spermatogenesis occurs through adolescence and adulthood. Sperm are produced continuously with the prolonged interruption of oogenesis. So another way you can look at this is that eggs are metabolically expensive for a female to produce, but sperm are metabolically cheap. So you can draw your own conclusions from that. The interplay of tropic and sex hormones regulates reproduction in mammals. Human reproduction is coordinated by hormones from the hypothalamus, anterior pituitary, and gonads. Gonadotropin-releasing hormone is secreted by the hypothalamus and directs the release of follicle-stimulating hormone and luteinizing hormone from the anterior pituitary. Follicle-stimulating hormone and luteinizing hormone regulate processes in the gonads and the production of sex hormones. The major androgen is testosterone, that's the major male hormone, and the major female hormone, or estrogen, is estradiol and progesterone. Both males and females produce androgens and estrogens, but differ in their blood concentrations and particular hormones. For example, males have testosterone levels about 10 times higher than females. Sex hormones regulate gamete production both directly and indirectly. They serve many additional functions, including sexual behavior and the development of primary and secondary sex characteristics. Androgens are responsible for male vocalizations and courtship displays. For example, here the male anole lizard, Norops ortoni, extends his dewlap, a brightly colored skin flap beneath the throat. Testosterone is required in both male in the male, both for the dewlap to develop and for the anole to display it to attract mates and guard his territory. Follicle-stimulating hormone and luteinizing hormone act on different cells in the testes to direct spermatogenesis. Sertoli cells are found in seminiferous tubules. They respond to follicle-stimulating hormone by nourishing the developing sperm. Leydig cells are connective tissue between the tubules that respond to luteinizing hormone by secreting testosterone and other androgens, which promote spermatogenesis. Okay, Sertoli cells, let me get my act together here. Okay, what's happening here is that luteinizing hormone is being released into the testes, stimulating the production of testosterone. Testosterone aids or promotes spermatogenesis. When that spermatogenesis reaches a certain level, negative feedback is given to the anterior pituitary and the hypothalamus, which will reduce the amount of. So it's a negative feedback loop. Once there's enough sperm here, then it will feed back and reduce the amount of luteinizing hormone. The follicle-stimulating hormone functions in much the same way. It's released into the Sertoli cells. It increases spermatogenesis, and in addition to inhibin, a hormone that in males produces by the Sertoli cells, acts on the anterior pituitary gland to reduce follicle-stimulating secretion. So follicle-stimulating hormone and luteinizing hormone released by the anterior pituitary in response to gonadotropin GnRH, gonadotropin-releasing hormone secretion by the hypothalamus acts on different types of cells in the testes to direct spermatogenesis. Sertoli cells located within the seminiferous tubules respond to follicle-stimulating hormone by nourishing developing sperm. Leydig cells scattered in conductive tissue between the tubules respond to luteinizing hormone by producing testosterone and other androgens which promote spermatogenesis in the tubules. The two negative feedback systems control sex hormone production in males. Testosterone regulates blood levels of gonadotropin-releasing hormone, follicle-stimulating hormone, and luteinizing hormone through inhibitory effects of the hypothalamus and anterior pituitary. In addition, inhibin, a hormone that in males is produced by Sertoli cells, acts on the anterior pituitary gland to reduce follicle-stimulating hormone secretion. Together, these negative feedback circuits maintain androgen production at optimum levels. Two negative feedback mechanisms control sex hormone production in males. Testosterone regulates the production of gonadotropin-releasing hormone, follicle-stimulating hormone, and luteinizing hormone through negative feedback mechanisms. Sertoli cells secrete the hormone inhibin, which reduces follicle-stimulating hormones secretion from anterior pituitary. Females produce eggs in cycles. Prior to ovulation, the endometrium thickens with blood vessels in preparation for embryo implantation. If the embryo does not implant in the endometrium is shed in a process called menstruation. Hormones link in the 2 cycles of female reproduction. Changes in the uterus define the menstrual cycle, called the uterine cycle, and changes in the ovaries define the ovarian cycle. Okay, so there are 2 closely linked reproductive cycles in human females. Changes in the uterus are defined as the menstrual cycle, also called the uterine cycle, and menstrual cycle averages 28 days, although cycles vary ranging widely from 20 to 40 days. The cyclic events in ovaries define the ovarian cycle. Hormone activity links in 2 cycles to each other, synchronizing ovarian follicle growth and ovulation with the establishment of a uterine lining that can support embryonic development. This figure outlines the major events of the female reproductive cycles, illustrating the close coordination across different tissues in the body. Okay, so the main cycle here is, we're going to try to keep these all on one page here. This diagram is actually broken down, but I think it's actually more convenient here to keep it all on one page. The ovarian cycle begins with stage 1, up at the top here, with the release of the hypothalamus of gonadotropin-releasing hormone, which stimulates the anterior pituitary to secrete small amounts of follicle-stimulating hormone and luteinizing hormone. That's section 2 here. Now section 3, follicle-stimulating hormone, as its name implies, stimulates follicle growth, aided by luteinizing hormone. And 4, the cells of the growing follicles start to make estradiol. This is where the slow rise in estradiol secreted during most of the follicular phase, the part of the ovarian cycle during which follicles grow and oocytes mature. Several follicles begin to grow within each cycle, but usually only one matures and the others disintegrate. The low levels of estradiol inhibit the secretion of the pituitary hormones, keeping the levels of FSH and LH relatively low. During this portion of the cycle, regulation of the hormones controlling the reproduction closely parallels the regulation in observed males. Okay, so this is what's happening in the ovarian cycle here. This is kind of this half of the left hand side is like the production of is like the first 14, 15 days of the menstrual cycle, and this half is the last 14, 15 days of the menstrual cycle. So this is what's happening here. This is where this is your time scale on the bottom here, and this is where ovulation is the main point here, where these hormones are being stimulated. Okay, when estradiol, this is part number 5. When estradiol secretion by the growing follicle begins to rise steeply, then we go to number 6. The follicle-stimulating hormone and luteinizing hormone levels increase markedly, whereas a low level of estradiol inhibits the secretion of pituitary gonadotropins, and high concentration has the opposite effect. It stimulates the gonadotropin secretion by acting on the hypothalamus to increase its output of gonadotropin-releasing hormone. The effect is greater for luteinizing hormone because the high concentration of estradiol increases the gonadotropin-releasing hormone sensitivity of luteinizing hormone releasing cells and the pituitary. In addition, follicles respond strongly to luteinizing hormone. Okay, where's 6? Okay. At this, they respond strongly to luteinizing hormone at this stage because most of their cells have receptors for this hormone. The increase in luteinizing hormone caused by the increase in estradiol secretion from the growing follicle is an example of a positive feedback. This result is the final maturation of the follicle here in section 7 on day 8 or 10. The maturing follicle containing fluid-filled cavity enlarges, forms a bulge near the surface of the ovary. The follicular phase ends at ovulation, about a day after the luteinizing hormone surge. In response to the peak levels in luteinizing hormone, the follicle and adjacent wall of the ovary rupture, releasing the secondary oocyte. There is sometimes a distinctive pain in the lower abdomen or at or near during the ovulation. This pain is felt on the left or right side, corresponding to whichever ovary has matured during that cycle. So it's interesting to note that ovaries and testes, they alternate in the production. So your left ovary will produce an egg, and then your right ovary will produce an egg. And your left testes will produce sperm, and your right testes will produce sperm. They alternate in phases. The luteal phase of the ovarian cycle follows ovulation. Okay, now we're back over to number 8. This is where the luteinizing hormone stimulates the follicular tissue left behind in the ovary to transform into a corpus luteum, a glandular structure. Under continued stimulation by luteinizing hormone, the corpus luteum secretes progesterone and estradiol, which in combination exert negative feedback on the hypothalamus and the pituitary. This feedback reduces the secretion of luteinizing hormone and follicle-stimulating hormone to very low levels, preventing another egg from maturing when a pregnancy may already be underway. Okay, so prior to ovulation, ovarian steroid hormones stimulate the uterus to prepare for support of an embryo. Estradiol secreted in increasing amounts by growing follicles signals the endometrium to thicken. In this way, the follicular phase of the ovarian cycle is coordinated with the proliferative phase of the uterine cycle. After ovulation, in section 9 here, The estradiol and progesterone secreted by the corpus luteum stimulate maintenance of the uterine lining, as well as the further development, including the enlargement of arteries and the growth of endometrial glands. These glands secrete a nutrient fluid which can be early embryo before it implants into the uterine lining. Thus, the luteal phase of the ovarian cycle is coordinated with what is called the secretory phase of the uterine cycle. And number 10, down at the end of our phase here, once the corpus luteum has disintegrated, the rapid drop in ovarian hormone levels causes arteries in the endometrium to constrict. Deprived of its circulation, the uterine lining largely disintegrates, and the uterus is in response to prostaglandin secretion contracts. Small endometrial blood vessels restrict releasing blood that is shed along with the endometrial tissue and fluid, and the resulting is menstruation, the menstrual flow of the uterine cycle. So this is how your hormones are related to ovulation and menstruation. Okay, so menstruation occurs right here, day 0. That's at the end of your cycle when your corpus luteum basically gives up, and then it becomes degenerating, and in its degeneration it no longer produces hormones necessary to maintain the uterine lining, and then the uterine lining sloughs off, and then the menstrual cycle begins again. Okay, here's the luteinizing hormone and follicle stimulating hormone. That luteinizing hormone precedes the ovulation. It's a little burst of luteinizing hormone, comes from the pituitary gland into the bloodstream. It encourages ovulation. That ovulation, it occurs just prior to estradiol release and progesterone. These progesterone and estradiol maintain the thickening of the endometrium, the lining of the uterus. When the estradiol, these are hormones that are related to the corpus luteum. When that corpus luteum eventually degenerates, it declines the level of luteinizing hormone and progesterone, and that signals to the uterus that it's no longer necessary to maintain the uterine lining, and the uterine lining then sloughs off during the menstrual phase. And then it happens all over again, every month. The sequential release of gonadotropin-releasing hormone and then follicle-stimulating hormone and luteinizing hormone stimulates follicle growth. Follicle and oocyte growth is an increase in the hormonal estradiol characterized the follicular phase of the ovarian cycle. Estradiol causes gonadotropin-releasing hormone, follicle-stimulating hormone, and luteinizing hormone levels to rise through positive feedback. The final result is the maturation of the follicle. At the end of the follicular phase, the follicle releases the secondary oocyte. The follicular tissue left behind transforms into the corpus luteum in response to luteinizing hormone. This is the luteal phase. The corpus luteum disintegrates and ovarian steroid hormones decrease by negative feedback mechanisms. Hormones coordinate the uterine cycle with the ovarian cycle. Thickening of the endometrium during the proliferative phase, it coordinates with the follicular phase. Secretion of the nutrients during the secretory phase coordinates with the luteal phase. Shedding of the endometrium during the menstrual phase coordinates with the growth of the new ovarian follicles. After about 500 cycles, the human females undergo a process called menopause. This is the cessation of ovulation and menstruation. Menopause is very unusual among animals, principally because most animals don't live into an older age. They're subject to natural predation and diseases and what have you. But humans have been able to live for such a long time that menopause is common among our species. It is unusual among animals, and menopause may have evolved to allow a mother to provide better care for her children and grandchildren. So it's possible that, I don't know, it just depends on your generation time. Some people have children when they're 18, 16 years old, and some people have children when they're 25, 30 years old. So I'm not sure. That's a fair amount of speculation. But at any rate, menopause does occur. It involves all kinds of hormonal shifts and changes. And if you have any relatives that have gone through it, you probably have heard about hot flashes and whatnot. Menstrual cycles are characteristic only of humans and some other primates. Endometrium is shed from the uterus in a bleeding called menstruation. Sexual receptivity is not limited to a time frame. Well, it's not. Humans don't become, like many species of animals, become fertile during a certain season, an estrus season, or season in which they are in heat. That could be like in the fall, so that they have young in the spring. But humans are not dependent on natural order in that way, and we can get pregnant in any season. So sexual receptivity is not limited to a particular time frame. Estrus cycles are characteristic of most mammals. The endometrium is reabsorbed by the uterus, and sexual receptivity is limited to a heat period or an estrus period. Different animals go through different periods of heat. Like, for example, river otters will go through heat in the fall, and then they can actually store that sperm. That they will not become fertilized until the springtime, and then they'll have their offspring. It will be a couple months after that, in the early summer. So the length and frequency of estrus cycles varies from species to species. Two reactions predominant in both sexes. There's vasoconstriction, which is the filling of tissue with blood, and myotonia, which is increased muscle tension. Okay, we're talking about human sexual response. The sexual response cycle has 4 phases: excitement, plateau, orgasm, and resolution. And Alfred Kinsey did a lot of the research on this. The Masters and Masters. There's all kinds of research that was done, like in the 50s and 60s, about human sexual response. There's even a movie about Kinsey and his experience studying human sexual response. Excitement prepares the penis and the vagina for coitus, sexual intercourse. Direct stimulation of genitalia maintains the plateau phase and prepares the vagina for the receipt of sperm. Orgasm is characterized by rhythmic contractions of the reproductive structures. In males, semen is first released into the urethra and then ejaculated from the urethra. In females, the uterus and the outer vagina contract. This will help to move the sperm into place into the fallopian tube. During the resolution phase, organs relax and return to their normal state. Okay. Fertilization, cleavage, and gastrulation initiate the embryonic development. So in this section, we're going to talk about the embryonic development, and we'll talk about this in a kind of a simplified manner, but it should give us a good overview. Across animal species, embryonic development involves common stages occurring at a set order. First is fertilization, which forms a zygote. During the cleavage stage, a series of mitosis divide the zygote into many-celled embryos. The resulting blastula then undergoes rearrangements into a 3-layered embryo called the gastrula. So here's an example of, looks like Xenopus laevis, the African clawed frog. Sperm here fertilizes the zygote. Then there undergoes an initial phase of cleavage where the cells first undergo mitotic division. Then it gets into even more developed mitotic division and called the blastula. The blastula becomes the gastrula, which is increasing development. You've got an ectoderm and an endoderm. You've got different parts of the embryo developing here. And then you have organogenesis into the embryo. And this embryo is the what remains inside the egg until it hatches out. When it hatches out, it becomes the larva stage cells. So this embryonic development is the same for pretty much all vertebrates, and even invertebrates, as we'll see here shortly when we look at the sea urchin egg. Okay, fertilization. Molecules and events at the surface of the egg surface play a crucial role in each step of fertilization. Sperm penetrate the protective area around the egg, and receptors of the egg's surface bind to protein molecules on the sperm surface. This changes the egg surface and prevents polyspermy, the entry of multiple sperm into a single nuclei in an egg. Okay, so here is the sperm. It's approaching the egg. Here's the acrosome, which contains the enzymes that help to dissolve the membrane, the surface membrane of the egg. This is the contact. The sperm contacts the egg's jelly coat, triggering exocytosis of the sperm's acrosome. This is the sperm acrosome being released here. Then the acrosomal reaction. The hydrolytic enzymes released from the acrosome make a hole in the jelly coat, and growing actin filaments form the acrosomal process, which protrudes from the sperm head and penetrates into the jelly coat. The proteins on the surface of the acrosomal process bind to receptors in the egg plasma membrane. So these actin filaments are extruded from the head of the sperm here, past the enzymes which are dissolving the jelly coat, and they're bringing particular proteins into these receptors. And once these receptors are activated, the contact and fusion of the sperm and egg membranes. The fusion triggers depolarization of the membrane, which acts as a fast block to spermatization. So within a couple of seconds, the polarity is changed in the membrane. This prevents the other sperm from approaching. And then finally, there is a cortical reaction where these receptors that have been stimulated, they shear off, and they allow this fertilization envelope to occur, and then this produces the slow block to polyspermy. So cortical granules in the egg fuse with the plasma membrane, and the secreted contents clip off the sperm binding receptors and cause the fertilization envelope to block. This is the slow block to fertilization. And then okay, mission accomplished. The sperm nucleus then enters into the egg. So you can see through this process, the first sperm that gets there fertilizes the egg, and in the process of fertilization, it allows these cortical granules to release. This then blocks any additional sperm from trying to fertilize the egg. The events of fertilization also initiate metabolic reactions that trigger the onset of embryonic development, thus activating the egg. Activation leads to events such as increased protein synthesis that precede the formation of the diploid nucleus. Sperm entry triggers a release of calcium, which activates the egg and triggers the cortical reaction, that slow block to polyspermy. Okay, so within a second, the binding of the sperm to the egg, the acrosomal reaction, within 10 seconds, increased intracellular calcium level. Within 20 seconds, cortical reaction, the slow block to polyspermy. And then within a couple of minutes, fertilization. Within a minute, fertilization forms. Increased protein synthesis in 5 minutes. In 20 minutes, the fusion of egg and sperm nuclei are complete. In 40 minutes, the onset of DNA synthesis. And by 90 minutes, you've got the first cell division. So fertilization occurs, the whole process occurs within an hour and a half of initial fertilization. Cleavage and gastrulation, these are processes describing the development of the embryo. Fertilization is followed by cleavage, a period of rapid cell division without growth. Cleavage partitions the cytoplasm of one large cell into many smaller cells. The blastula is a ball of cells with a fluid-filled cavity called the blastocele. The blastula is produced after about 5 to 7 cleavage divisions. Okay, here's a fertilized egg. Looks like this could be a mammal egg. The egg gets fertilized. The initial blastula stage is the 4-cell stage. Then there's a 16-cell early blastula phase, and then a later blastula phase. Here's the ectoderm and the endoderm, and the mesoderm is going to form in there. And we're going to take a closer look at that in this next couple of slides. So shown here is the zygote shortly before the first cleavage division, surrounded by fertilization envelope. This is the 4-stage cell. Okay, well, these are all pretty self-explanatory. Cleavage is a series of mitotic cell divisions that transform a zygote into a blastula, a hollow ball of cells called blastomeres. The light micrographs show that the embryonic stages of the shell of a sand dollar, which are virtually identical to those of a sea urchin. After cleavage, the rate of cell division slows. The remaining stages of embryonic development are responsible for morphogenesis, the cellular and tissue-based processes by which the animal body takes shape. After gastrulation, a set of cells at or near the surface of the blastula moves into the interior location. These cell layers are established, and a primitive digestive tube forms. The hollow blastula is reorganized into a 2 or 3 layered embryo called the gastrula. The cell layers produced by gastrulation are called germ cell layers. The ectoderm forms the outer layer and the endoderm the inner layer. In vertebrates and other mammals with bilateral symmetry, a third germ layer, the mesoderm, forms between the endoderm and the ectoderm. Okay, here is the sea urchin embryo. This is the sea urchin embryos are classically used because they are easy to see and they are deuterostomes. Remember, deuterostomes are include the echinodermata and the chordates. So chordates gave rise to vertebrates. So vertebrates, they have the same type of embryonic development. The anus forms from the first opening. So deuterostome means second opening. The anus is here is the first opening, and the mouth is the second opening. So that's the deuterostome. Okay, just like us, echinoderms and chordata. Okay, the movement of the cells during gastrulation forms an embryo with a primitive digestive tube and germ layers, and 3 germ layers. Some of the mesenchyme cells that migrate inward, step 1, will eventually secrete calcium carbonate and form a simple internal skeleton. Embryos in steps 2 and 3 are viewed from the front, those in steps 4 and 5 from the side. Okay, let's see. So the first part up here at the top, once the blastula has formed, the gastrulation begins with the migration of mesenchyme cells from the vegetal poles into the blastocele. So here's the blastocele inside, and here's the mesenchyme cells. They're eventually going to become the mesoderm. The vegetal state invaginates. Okay, it begins to get a hole into it. The mesenchyme cells migrate through the blastocele, and stage number 3, the endoderm cells form the archenteron, future digestive tube. New mesenchyme cells at the tip of the tube send out thin extensions, filopodia, towards the blastocele wall. The filopodia then contract, dragging the archenteron across the blastocele. And 5, finally, the fusion and the archenteron with the blastocele wall forms a digestive tube, which now has a mouth and anus. The gastrula has 3 germ layers and is covered with cilia, which function in later in feeding and movement. Okay. That's just a review. This is on page 745. The ectoderm is the outer layer of the embryo. The mesoderm is in red. That's the middle layer of the embryo. And the endoderm, that's the inner layer of the embryo, the part that's going to have the lumen that will construct the digestive tube. Cell movements and interactions that form the germ layers vary among species. One distinction is whether the mouth develops at the first opening that forms the embryo in protostomes or the second in deuterostomes. Sea urchins and other echinoderms are deuterostomes, as are chordates. Each layer contributes a distinct set of structures in the adult animal. Some organs and many organ systems derive from more than one germ layer. There's the ectoderm, which provides the epidermis, the nervous and sensory systems, pituitary glands, adrenal medulla, jaws and teeth, germ cells. The mesoderm, which provides the middle layer, which is the skeletal and muscular systems, the circulatory and lymphatic systems, the excretory and reproductive systems, except for the germ cells, the dermis of skin, and the adrenal cortex. And then there's the endoderm, the innermost layer of the embryo, which gives rise to the epithelial lining of the digestive tract and the associated organs, the liver and pancreas, and the epithelial lining of the respiratory, excretory, and reproductive tracts and ducts, thymus, thyroid, and parathyroid glands. Embryonic development and birth. Conception is the fertilization of an egg by a sperm. It occurs in the oviduct, what I've been calling the fallopian tube. The resulting zygote begins to cleavage in about 24 hours after fertilization and reproduces a blastocyst after about four more days. a few days later, the embryo implants into the endometrium of the uterus. The condition of carrying one or more embryos in the uterus is called gestation or pregnancy. The pregnancy averages 38 weeks from fertilization for humans. Gestation varies among mammals from 21 days in many rodents to more than 600 days in elephants. Human gestation is divided into 3 trimesters of about 3 months each. Okay, here, let's start down here at stage 1, ovulation. Ovulation releases the secondary oocyte, which travels into the oviduct. Okay, you can see there's kind of like a little bit of space right here, and this is where, if anybody is familiar with the phrase "getting your tubes tied" or tubal ligation, what happens is they tie off or remove a section of the fallopian tube here, and then the eggs just get released and they just kind of float around in this area around the uterus. They don't find their way down the fallopian tube and into the uterus, and that's a form of contraception. Okay, so anyway, ovulation releases the egg and secondary oocyte, which travels into the oviduct, the fallopian tube. There's fertilization occurs as a sperm enters the oocyte. Meiosis of the oocyte is completed, and the nuclei of the oocyte and the sperm fuse, producing the zygote here in the fallopian tubes. Cleavage is the cell division. It begins in the oviduct as the embryo moves toward the uterus by peristalsis and movements of the cilia. So remember peristalsis when we talked about the digestive tract? A similar effect is occurring here in the fallopian tube. 4, cleavage continues. By the time the embryo reaches the uterus, it is a ball of cells and floats in the uterus for several days. Nourished by the endometrial secretions, it becomes a blastocyst. And then the blastocyst itself implants into the endometrium about 7 days after conception. Okay, here is the implantation of a blastocyst into the uterine lining. Once it's implanted, it becomes there is the region known as the trophoblast, which then becomes the placenta. This is the area that is going to attach through the umbilical cord into the fetus. During the first trimester, the embryo secretes hormones that signal its presence and regulate the mother's reproductive system. Human chorionic gonadotropin acts to maintain secretion of progesterone and estrogens by the corpus luteum through the first few months of pregnancy. Occasionally, an embryo splits during the first month of development, resulting in identical or monozygotic twins. Fraternal or dizygotic twins arise when 2 eggs are fertilized and implant as 2 genetically distinct embryos. Some embryos cannot complete development due to chromosomal or other abnormalities, and many times these are spontaneously aborted, often before a woman is even aware of her pregnancy. During the first 2 to 4 weeks, the embryo obtains nutrients directly from the endometrium. Meanwhile, the outer layer of the blastocyst, called the trophoblast, mingles with the endometrium and eventually forms the placenta. Materials diffuse between maternal and embryonic circulation to supply the embryo with nutrients, immune protection, and metabolic waste disposal. The first trimester is the main period of organogenesis, the development of the body organs. All of the major organs are present by 8 weeks, and the embryo is called a fetus. Okay, by 5 weeks, the limb buds and eyes and heart and liver and rudiments of all other organs have started to develop in the embryo, which is only about 1 centimeter long. By 14 weeks, growth and development of the offspring, now called a fetus, is continued during the second trimester. This fetus is about 6 centimeters long. By 20 weeks, this fetus has grown nearly 20 centimeters in length and requires adoption of the fetal position, head to knees, and due to the limited space available. So it's interesting here. You can see here's the umbilical cord. It's like almost as big as the fetus here, and then here's the umbilical cord here attached to the placenta. This is the placenta, which is attached. The placenta is then again attached to the endometrial lining, and here's a little baby all crammed up inside there. For the first 6 weeks of development, the embryo has either XX or XY chromosomes, but physically there is no differentiation between males and females during the first 6 weeks of gestation, and this is why men have nipples, because we're basically all the same, especially during the first 5 weeks of development. So we have all the same body structures. There's no real differentiation. There's no differentiation of the sex organs until you get much older. Well, not much older, but until you get older. Okay, here's the 5 week. That's about 1 centimeter long. And 14 weeks, that's about 6 centimeters. And 20 weeks, that's about 20 centimeters. During the second trimester, the fetus grows and is very active. The mother may feel fetal movements as early as 1 month into the second trimester. During the third trimester, the fetus grows and fills the space with embryonic membranes, within the embryonic membranes. Childbirth begins with labor, a series of strong rhythmic contractions that pushes the fetus and the placenta out of the body. And once labor begins, local regulators, prostaglandins, and hormones induce and regulate further contraction of the uterus. Lactation, the production of milk, is an aspect of maternal care unique to mammals. Suckling stimulates the release of prolactin, which in turn stimulates mammary glands to produce milk, and the secretion of oxytocin, which triggers milk release from the glands. Okay, there are many different kinds of contraceptions. I think we're going to go through a few here, but I think the most reliable and easiest to use is the condom. And anyway, we'll talk about them here. Contraception is a deliberate prevention of pregnancy, which can be achieved in a number of ways. Contraceptive methods fall into 3 categories: preventing development of release of eggs and sperm, keeping sperm and eggs apart, and preventing the implantation of an embryo. So that's what keeping sperm and eggs apart is like with a barrier, with the condom, and preventing implantation of the embryo. That's like what the pill does, the birth control pill. Healthcare providers should be consulted for complete information on the choice and risks of contraception methods. Everybody has their own preferences, and it's something that deserves a little research. Okay. All right. I don't know what we got here. Here's a table. Red arrows indicate where these methods or devices or products interfere with events from production of sperm and primary oocytes to an implanted in production embryo. Okay, so a vasectomy is when you cut the vas deferens. The analogous thing would be tubal ligation, tying the tubes, which prevents the oocyte from being released into the uterus. Abstinence, okay. Obviously, abstinence works if you practice it. It's not always easy to be in a relationship and practice abstinence. Coitus interruptus, that's the withdrawal method. It's not. Look, I'm not going to really go over this. There's all different kinds of contraceptive methods, and everybody here is of an age where I'm sure they're familiar with these different potentials. So if you want to learn more about that, that's on page 748 in your textbook. Okay, then there's all right, the rhythm method or natural family planning, which is when you have sexual relations at a time when you're least likely to get pregnant. Coitus interruptus is the withdrawal before ejaculation. It's unreliable. Barrier methods block fertilization when pregnancy rate has less than 10%. Okay, intrauterine devices, pregnancy rate is less than 1%. They set up like a small infection on the inside of the uterus, and this prevents the egg from implanting. And female birth controls are hormonal contraception with a pregnancy rate of less than 1%. So birth control pills are pretty good, but it does mess around with your hormonal cycling, so it may not be desirable, or it may be desirable. It depends on what your preferences are. Okay, then there's different types of fertility treatments. Infertility is the inability to conceive offspring. It affects 1 in 10 couples in the United States and worldwide. The causes are varied and equally likely to affect men and women. There are sexually transmitted diseases are the most significant preventable causes of infertility. So, like gonorrhea, chlamydia, can lead to infertility. Some forms of infertility are treatable. Hormone therapy can sometimes increase sperm or egg production. In vitro fertilization mixes oocytes with sperm in culture dishes and returns the embryo to uterus at the 8-cell stage. The sperm may be directly directed into an oocyte as well. So there's different ways of doing in vitro fertilization. Okay. This is an experiment where they took rabbits and they cut off their genitals as they were in the embryonic stage. They either developed, if they were XY, they developed into male. If they were XX, they developed into female. If they removed the gonads, then both they would all become female. So male gonads are necessary to produce the hormones required to develop the male animal. And this is also when I was talking about, like, in their first 6 weeks of embryonic development, we all have the same traits. We're basically all female, and that's why men have nipples. Fascinating topic there. Okay, here's gametogenesis, spermatogenesis. Look at that. The male is producing tons and tons of sperm. The female goes through a similar pathway, but only produces one egg. So the female, each egg is precious. The male, every sperm is not so much precious as the egg.